Welcome to today's topic. Why is third generation iPSC or induced pluripotent stem cells the best and most advanced stem cell therapy available to date? We will look at the advancement in stem cells applications and innovation through the lenses of third generation iPSC therapy. iPSC means induced pluripotent stem cells, and I'll explain it further what they are. First, is aging a disease? Can it be treated if it's a disease? Well, in the recent World Health Organization International Classification of Disease 11, aging is a medical target with two codes. So, inclusion of these two codes means that aging is treatable. In the current ICD-11, there's a code MG2A under causality. Old age is now fully replaced with a new one, aging-associated decline in intrinsic capacity. Another code, XT9T, extension code aging-related in the causality or etiology category. Which also permits targeting aging as a medical indication, namely as an underlying cause or risk factor of diseases. So, with these two codes, aging can now directly be targeted by biomedical interventions, biohacking, whatever you want to call it, in accordance with the present international classification of disease. Okay, just to put things in perspective, we look at the stem cell continuum. When an egg and sperm fertilize, the first 3 days, it's called totipotent stem cells. Okay, from day 4 or 5 onwards to end of week 10 is the embryonic stage. Okay, and from week 11 until birth, it's the fetal stage. Okay, so the embryonic stem cells are pluripotent, whereas the fetal stem cells from week 11 onwards are more multipotent to maybe a bit pluripotent, and adult stem cells, of course, are multipotent or unipotent. So, iPSC can be autologous or it can be allogenic. Okay, mostly, of course, the best is coming from your own cells. Okay, it's getting your embryonic stem cells that you had when you were in your mommy's womb from day 4 to end of week 10, you know, that embryonic stage. So what people can do now is they get some of your adult cells and they can reprogram it to your own stem cells when you were in your mommy's womb from day 4 to end of week 10. Okay, because week 11 onwards to birth, it's a fetal stage, it's not the embryonic stage, and that's when you have multipotent stem cells. Potency of stem cells. Totipotent stem cells result from the fusion of an egg and sperm, can form any cell of the embryo as well as the placenta, can construct an entire viable organism. So basically, that's from day 1 to day 3 of fertilization. Pluripotent stem cells from day 4-5 to end of week 10. Pluripotent or embryonic stem cells, they can differentiate into nearly all cell types, cells derived from any of the 3 germ layers except the placenta. Multipotent stem cells, they can differentiate into a number of cells, but only those closely related to a family of cells, and that's probably from week 11 to end to birth of the child. That's the fetal stage, and that's where the cells are multipotent. For instance, hematopoietic stem cells in the bone marrow, they can only produce cells related to blood. Okay, like red blood cells, white blood cells, platelets, etc. And unipotent stem cells can produce only one cell type, but of their own. Like for instance, a heart stem cell can produce only heart cells, but they have the property of ability for self-renewal. It means they can make more of itself. They can make more. One heart stem cells can become 2 heart stem cells, or 4 and 8, etc., etc. Embryonic stem cells are pluripotent stem cells derived from the inner mass of the blastocyst. And here you go. Here's the oocyte, the egg. When it's fertilized, the first 3 days, it forms a morula, 4, 8, 16 cells. It's the first 3 days. By day 4, it forms the blastocyst. And inner mass is where you get your embryonic stem cells. There you go. Here, embryos reach the blastocyst stage 4 to 5 days post-fertilization. In the past, of course, people were paying millions to go to certain centers for embryonic stem cells. The human embryos are sacrificed. I mean, a potential life is sacrificed. So there are obvious ethical issues regarding its use. The potential use include diabetes, heart disease, spinal cord injuries, but the problem is that they form a lot of teratomas or tumors. So pluripotency refers to embryonic stem cells. They are able to differentiate into all 3 germ layers: your ectoderm, which forms your exoskeleton, your skin; your mesoderm forms your organs, your muscle mass, your bone; and endoderm forms the inner lining of your organs, like your mucosa, your lining of your intestinal tract. And these germ layers generate each of the more than 220 cell types in the human body. When provided appropriate signals, embryonic stem cells initially form a precursor cell, there you go, and they can subsequently differentiate into the desired cell types. Like if you need a heart cell, if you need a muscle cell, or you need a blood cell, they can be induced to form that particular cell type. For instance, if you had a stroke, you can get your adult stem cells from your blood or your skin, and we can reprogram it to form your own embryonic brain cells, right? When you were in your mother's womb, day 4 to week 10. And they're capable of cell renewal. So in addition to human embryonic stem cells there has been research focused on induced pluripotent stem cells to bypass the controversy and ethical issues using embryonic stem cells, of course, right? Because you're killing an embryo. The iPSC technology was pioneered by Shinya Yamanaka's lab in Kyoto, Japan. He showed in 2006 4 specific genes encoding transcription factors could convert adult cells into pluripotent stem cells, right? So using 4 genes, he can convert your adult cells to your embryonic stem cells when you were in your mother's womb, day 4 to week 10 of pregnancy. And he was awarded the Nobel Prize in 2012, along with John Gurdon, for the discovery that mature cells can be reprogrammed to become pluripotent. So the capid-induced pluripotent stem cells are derived from skin or just 20 to 50 mL of your blood. They can reprogram it back into embryonic-like pluripotent stem cells that enable the development of unlimited source of any types of human cells needed for therapeutic purpose. So we can reprogram back your adult cells back into the embryonic stem cells when you were in an embryonic state in your mother's womb, right? So iPSC bypassed the needs for embryos in stem cell therapy. To produce cell therapy, somatic cells are recovered from the patient, from your blood, for instance, or your skin. Just for autologous therapy, it means using your own cells, or from immune-compatible donors as allogenic therapy. So obviously preferable is using your own autologous, followed by reprogramming to an undifferentiated states using small molecules. And there are 4 small molecules that were initially used. There you go. These are called transcription factors. So Yamanaka used these 4 transcription factors: OCT, OCT34, SOX2, KIF4, and CMYC, right? And then they add it to your adult cells, and they become pluripotent stem cells, right? And they can differentiate into whatever cells is needed. Say if you had a heart, you need heart cells, they can be reprogrammed to make your own embryonic heart cells. Or if you had you have diabetes, you need a new pancreas, they can be programmed back to produce your pancreatic cells when you were in your mummy's womb, right? I mean, you differentiate back. So that is the transcription factors.
Multicellular organisms, such as a cat, have many billions of cells. These include somatic cells, shown here in orange, and stem cells, shown here in green. Somatic cells, from the Greek soma or body, make up the body of an organism and are committed to a specific cell fate. For example, the skin cell can divide to make more skin cells, but not muscle cells or other types of cells. In contrast, pluripotent stem cells, pluri means several, while potent means to be able, can make any kind of cell. For example, both skin and muscle cells. Pluripotent stem cells exist as part of normal development embryos, but they do not exist in adults. So is it possible to give adults pluripotent stem cells? In 2012, Dr. Shinya Yamanaka won the Nobel Prize for discovering that adding the 4 genes MYC, OCT3 or 4, SOX2, and KLF4 into somatic cells leads to induced pluripotent stem cells, or iPSCs. Why is this so important in medicine? Because iPSCs can grow continuously and give rise to every other cell type in the body. They can be used to replace damaged cells and tissues, such as in the case of muscle damage, contributing to the field of regenerative medicine. Since iPSCs can be derived from adult somatic cells, each individual patient could have their own stem cell line for organ transplants, for example, to get a new liver. In addition, iPSCs can be used to test what specific treatment works best for individual patients, contributing to the field of personalized medicine.
So just to cap it, they use these 4 genes, they add to your adult stem cells, and they are able to make iPSC cells, liver, your spinal cord cells, whatever. So they can program back to make your own embryonic stem cells. Okay. So iPSC have the ability to expand infinitely. As the saying goes, 1 gives birth to 2, 2 gives to 3, and etc. And the seeds are all reusable. And iPSC are scientifically stored at ultra low temperatures, 196, and revived regularly so they can be preserved permanently, achieving immortality at the cellular level. That means they can be stored safe permanently, right? Should you need it, say 10 years down the road, 20 years down the road, or even 100 years down the road, they can be safe permanently. And they can iPSC can be differentiated into all cell types, including neurons, cardiomyocytes, your heart cells, liver cells, blood cells, and more. Okay, they can replace any cells in your body. Personalized iPSC is an inexhaustible and everlasting life bank. In the past, a lot of people, when they give birth, they collect the cord blood stem cells of the newborn. Okay, what's its potency? It's fairly low. Does it need matching? If it's the own child using their own umbilical cord, yeah, then there's probably no need for matching. But if you use it on another person, you need to match it. Okay, and usually stored for at most 15 to 20 years. After that, there's no potency anymore. And medical and health values only enough for children under 8 years old with leukemia, etc. Some people collect their immune cells and they store it. Okay, it's again, you can only store it for about 10 years to 15 years, and it's to prevent and treat tumors and viral disease, and it improves immunity. Like you can collect your own natural killer cells and so forth. Adult stem cells. I mean, there are some people who has collected their adipose stem cells and then they've stored it. Okay, potency middle, self-use, and again, you probably store it for about 10 to 20 years. Wide range of use, but more like for general conditioning to make yourself younger, but there's not much therapeutic use, right? Because you can't really differentiate it into specific organ type. iPSC, blood collection, 20 to 50 mils, is pluripotent, self-use, because it can be autologous, your own. It can store unlimited time, it can be permanently stored, and the whole full spectrum of disease can be treated because it can be used to differentiate into all cell types and all organ types. This is a center in Shanghai that offers iPSC. I've personally visited it. If you need any more information, people who need treatment for general well-being or spinal cord injuries or Alzheimer's or dementia, stroke, any serious chronic conditions, do give me a thinker. I can be linked to this center for treatment. And again, in comparison with, say, the first generation iPSC, they use the 4 programming factors, but they cause a lot of tumors. Okay, so they remove the c-MYC, which cause a lot of tumors, and that's basically second generation iPSC. But the differentiation control was still not as good. Okay, they were using viral vectors, plasmid vectors. They're using lentivirus, sendivirus, and so forth. So the differentiation control was not as good. So the third generation iPSC is the most advanced. They use microRNA and mRNA for differentiation control, so that you can control the cell types you want much more better. So they don't use these viruses anymore as in the reprogramming phase. Embryonic stem cells we talked about, ethical issues, and a lot of tumors caused. Mesenchymal stem cells are usually not your stem cells, right? They're allogenic rather than autologous, and the limited usage. Okay, I won't go into it. So advancement, this presentation dwells into the advances of third generation induced pluripotent stem cell therapy, examining its definition, which I've done, right? Advantages over earlier generations and the current applications in research and medicine, which I will now share. Third generation induced pluripotent stem cells represent a significant advancement in stem cell technology, allow for direct reprogramming of somatic stem cells into pluripotent state without the use of harmful factors such as viruses. So the first 2 generations, they use viruses in the reprogramming state. Okay, now in third generation, they don't. They use microRNA and mRNA. This generation utilizes safer reprogramming methods, enhancing the clinical and potential usability of iPSC in therapeutic conditions. So it can treat disease rather than just making you beautiful, making you look young. Okay. Advantages over previous generations. The primary advantage of third generation iPSC include improved safety profiles due to non-integrating reprogramming strategies, enhanced efficiency in generating stable and functional cells, and reduced risk of tumors, right? Like, especially the first generation iPSC. There were a lot of teratomas, tumors. These advancements enable more reliable patient-specific therapies and broader applications in regenerative medicine, while also addressing ethical concerns associated with embryonic stem cells. So you don't need to kill or sacrifice the life of an embryo like you do with embryonic ESC therapy early on. Current research and applications in third generation iPSC focuses on various applications, including cell replacement therapies for degenerative disease, tissue reengineering, and personalized medicine. Studies are underway to understand the mechanism of differentiation and the potential for using these cells in transplantations, alongside ongoing trials for various conditions such as Parkinson's and spinal cord injuries. They can be reprogrammed to any cells in your body. Okay, if you need nerve cells, like you have a spinal cord injury, stroke, Alzheimer's, etc. They can be differentiated specifically into your nerve cells, muscle cells, organs, blood cells, bone cells. Okay. Clinical applications. In regenerative medicine, third generation iPSC have the potential to regenerate damaged tissues and organs. The ability to differentiate into various cell types allows for treatment of conditions such as heart disease, diabetes, and neurological disorders. I mean, they can be differentiated into specific cell types that's needed. Okay, if you have heart problems, they can be differentiated into cardiomyocytes, okay, heart cells. If you have diabetes, they can be differentiated into pancreatic islets cells into pancreas, okay, so to make your pancreas functional again, right? And likewise with neurological diseases. These cells can be derived from the patient's own tissues, minimizing the risk of immune rejection and enhancing treatment efficacy. Disease modeling. Third generation iPSC can facilitate the creation of accurate disease model that replicate patient specific conditions in vitro. This enables researchers to study mechanisms, identify potential drug targets, and evaluate the efficacy of new treatments in a controlled environment. Such models are invaluable for understanding complex disease such as Alzheimer's, Huntington's, and various genetic diseases. So if you have a genetic disease, iPSCs of your own cells can be used to see which treatment, which drug will be more effective, right? And so forth, or even which nutrients, which vitamins suits you better. So this is a study that I just found on the internet and Parkinson's website using iPSC for phase 1 trial. It's been approved in China. This is a iPSC stem cell trial for Parkinson's disease, approved for phase 1 clinical trials. This was just a few days ago, this news. So drug discovery and testing. The application of third generation iPSC in drug discoveries enhances the efficiency of testing pharmaceutical compounds. By using patient-derived iPSC, researchers can now screen new drugs on cells that mimic the patient's own biology. This personalized approach allows for more accurate prediction of drug response and reduces the likelihood of adverse effects during clinical trials. So in conclusion, the advancement in third generation induced pluripotent stem cells therapy holds significant promise for the future of medicine. Their unique properties enhance safety, effectiveness, and personalization of treatments, particularly in regenerative medicine, disease modeling, and drug discovery. So thank you for listening.